and it's a pleasure to present my doctoral work about FAZ10 protein in Trypanosoma brucei. Trypanosoma brucei is a parasite causative agent of this disease, sleeping sickness. We can see the parasite with erythrocytes in humans and people with this disease. This disease is localized in the sub-Saharan Africa. The parasite has a crucial flagellum attachment zone. This is called also FAZ. And this zone is very important for cell morphology and survival of this parasite. This FAZ is a complex of protein interacting between them. One of these proteins is a FAZ10, the focus of my project. This protein is very important in cytokinesis. This young protein with a high molecular mass of 2 million Daltons. The principal objective of my work is the central region. We know that this central region has a coil domain that is very important for oligomerization state. We can see here this flagellum attachment zone between the flagellum and cell body. The microscopic image showing this interaction between those components and the localization of FAZ10 protein. We can see here this FAZ10 divided into 5 regions for better study. We can see here the central region. We have focused in this region. Why? Due to the information about the sorting of this protein. We have performed the sorting prediction, and we have selected the central region because it's more structured, or it has low disorder. So we have considered like an initial point of the structural studies of the FAZ10. We have performed a secondary structure prediction and 3D structure prediction with the famous tool AlphaFold 2 and coiled coil analysis. Experimental analysis was with cloned sequencing and transformation. We have made some formatted clones, had made expression and purification of recombinant proteins with affinity chromatography and size exclusion chromatography. And we have stabilized different protein concentration according to the next steps. Segmals, circular diachorism, and negative staining. The results present you the dimer model of the central region, the coiled coil domain of 44 nanometers of length, flanked by 2 globular domains. About the coiled coil analysis, in sequence we can see in red the classic hydrophobic amino acid in the position A and D. In this case, leucine. This amino acid has the responsibility of the interaction between both chains. And in the position A and G, in yellow, we can see the charter residues. These residues have the function to support the interaction of the amino acid, hydrophobic amino acid. And we have identified this classic knob into hole. The knobs, in the case of amino acid A and D, into this hole composed by 4 amino acids. About the experimental results, we have obtained 3 recombinant proteins. The first one in black, all the central region. The second one, the coiled coil domain in blue, and in red, just the globular domain. The central region with 66 kilodaltons, the coiled coil domain with 35 kilodaltons, and the globular domain just with 12 kilodaltons. But to know about the real molecular mass of this recombinant protein, we have performed the segmals, and we have found that the central region in black is presenting 124 kilodalton. That it's a dimeric conformation. In blue, the similar situation with the coiled coil domain that it's present 55 kilodaltons. And in contrast to the globular domain in red with 12 kilodaltons. It means that the central region has a dimer conformation due to the presence of the coiled coil domain. And to know that is if the theoretical or in silico result and experimental result are talking the same thing, we have performed a circular dichroism, and we have found the similar minimum value in the case of central region in both results A and B, and the globular domain in both results C and D. It means that both sets of results have the same characteristic. And finally, we have performed a negative stain with electron microscopy, and we have found, indicated with yellow arrows, filamentous structure with 50 nanometers, corresponding to the previous in silico prediction, either linear or V-shaped. In conclusion, the central region of FAZ10 have a dimeric conformation with one coiled coil flanked by two globular domain, and this filamentous structure it consisted with this scaffold-like role of the FAZ10 within the FAZ. If the FAZ10, the central region of FAZ10 is a dimer, we understand that all this FAZ10 is also a dimer. We have made a validation of this in silico prediction with experimental analysis. It means the relevance, the importance of in silico prediction to understand more about the function of relevant proteins in Trypanosoma brucei in this case, and it could be applied in all the organisms that you want to study. Thank you.